It's my pleasure to welcome Louise Purton, who's the head of the Stem Cell Regulation Unit at St. Vincent's Institute, and is also the Assistant Director there. Louise completed her PhD across the road at the Uni of Melbourne, working on the role of the stem cell bone marrow microenvironment in megakaryocyte development. And she continued that theme when she went to the Fred Hutchinson Cancer Research Centre in the late 90s, where she worked on stem cell mobilisation and differentiation, the role of the microenvironment in that, and with a particular focus on vitamin A or retinoic acid and its receptors and the role that they played. She returned to Australia to the Peter Mac in the early 2000s, she continued work on the role of retinoic acid receptors and the microenvironment on stem cell development, and continued that as well when she was a visiting scientist at the Massachusetts General Hospital from 2004 to 2007. And there she made a number of key discoveries, including the seminal one that deletion of retinoic acid receptor gamma causes a microenvironment-induced myeloproliferative disorder, and that was really the first indication that the microenvironment could cause myeloproliferative disorders. So it was a very interesting discovery. And she returned to the St. Vincent's Institute, where she's now a world-renowned authority on stem cells and the role of the microenvironment, and is invited speaker at just about every international conference I go to. Despite that, I still look forward to every one of her seminars, and I look forward to this one. Thanks, Louise.
Okay, thanks, Matt. So I'd like to thank Matt and John and Ruth for inviting me, and it's really great to see a lot more interest coming out in the microenvironment, which is an area of research I'm incredibly fond of. So I want to give you a basic introduction both to haematopoiesis and to the microenvironment. And for those of you who don't study haematopoiesis or don't really appreciate how dynamic it is, this is a slide that actually got me hooked on haematopoiesis when I was an undergraduate student. And basically my supervisor, who was the lecturer, pointed out that we make billions of blood cells every day. So it's a very dynamic system. And as a result of this, there's also a lot of complications for cancer patients, and I'll get to that in a bit. So we need normal numbers of mature blood cells produced every day. We need the erythrocytes, which are coloured in red here, to carry the oxygen around the body and protect us against hypoxia. We need the platelets, which are the tiny little ones down here, to heal the body after bleeding. So they're the first defense in clotting up the wound. And then we need the different types of leukocytes to help fight infection. And we know that there's actually going to be a few studies suggesting that maybe we've got it a bit wrong here, that there's actually a hierarchy of blood cells. The long-term repopulating stem cell, which is actually considered to be the only cell that can repopulate long-term in mice and in humans, and I'll also get to some data later proving that maybe this is not quite the case after all. Then they undergo a bit of differentiation, become short-term repopulating stem cells, then the multipotent progenitors, and they branch out to the lymphoid progenitors and the myeloid progenitors, and ultimately they form hematopoietic cells. So when we have changes occurring in any of these cell types along the way, we can get different types of blood cell diseases or blood cell cancers. Okay, so microenvironment. This is basically a timeline that David Scadden recently published in Cell Stem Cell. It's quite a nice review. There are some things that he's missed out on when he's been in our seminal Cell papers in 2007, which he was actually an author on my senior author paper, but anyway. But there are actually a couple of others that I think are more important and some of my favorites. So in the early to late 1960s, the discovery of the Steele-Dickey and WWV mutant mouse actually provided some interesting insight into haematopoiesis and how it's regulated. So at the time, they really didn't know what the differences between these mice were, but they knew that they both got macrocytic anemia and they shared a lot of different features. However, transplantation studies about wild-type marrow into the Steele-Dickey or the Steele-Dickey marrow into wild-type mouse, and the WWV into wild-type mouse or wild-type mouse marrow into WWV recipients, revealed that the WWV hematopoietic defect was cell-intrinsic, and the Steele-Dickey defect was a microenvironment defect. And then later on, it turned out that the Steele-Dickey have a defect in KIT ligand production, and the WWV have a defect in the KIT receptor on the cell surface of the hematopoietic cell. So really, in my opinion, this is the first evidence of the microenvironment having essential roles in hematopoiesis. It's a landmark group of studies. And then another favorite study, and I'm going to show you a little bit of data from this, is John Trenton and Norm Wolf. And Norm Wolf was actually trained as a veterinarian. And they did elegant studies, which I'll get to soon, where they transplanted spicules from the bone marrow cavity, which contained bone, into irradiated spleens. And they could prove that there were different microenvironments supporting hematopoiesis. I'll explain that in a little bit in a minute. And then Leon Weiss, who in the 1960s to the 1980s did a lot of very elegant transmission electron microscopy and scanning electron microscopy studies, revealing that there are multiple different types of microenvironments in the bone marrow. It's not just about the stem cell, which has become the big thing, but any of these microenvironments are essential, and if you disrupt them, you can perturb hematopoiesis. So there are also some favorite ones of mine. Okay. So specialized microenvironments also exist in the thymus. And I must admit, I thought I was going to show you a bit more data from our lab from what we've done, but I don't have time, so let's just summarize briefly what we've done here. But what is known is that there are these cortical epithelial cells here, and they're actually bright for CDR1, and they also express EpCAM. And they're important in the generation and the positive selection of double-negative thymocytes. Then we have the medullary epithelial cells. These are CDR1 low and EpCAM positive. And you can see them down here. And they're involved in the maturation and negative selection of single positive thymocytes. And then this population here, these CDR1 positive stromal cells, their function was not really known. We actually have a paper currently under review where we show that there's a good portion of these are nestin-expressing cells, and that they express high levels of CXCL12 and stem cell factor, more so than the EpCAM epithelial positive epithelial cells. And that when we delete REA gamma in these nestin-expressing cells, there's no impact on stem cells, but we get reductions of pre-B cells in the bone marrow, which is known to contain nestin-expressing cells, and we get a reduction in the DM4 T cells in the thymus. And when we treat wild-type mice with ATRA, we get the opposite effect. So we get increased pre-B cells in their marrow, and we get increased DM4 T cells. Okay, so that's what I'm going to say about the thymus, sorry. Specialized microenvironments also exist in the spleen, and this actually is the topic of one of my favorite microenvironment studies back from the 60s, before I was even born, actually. So we have the red pulp here, you can see that quite nicely, and then you get the white pulp, which is predominantly lymphopoiesis. So the B cells are maturing here in their follicles, and they're surrounded by some T cells as well. Normally the spleen, and I must say in the mouse, the spleen is always hematopoietically active, but in the human, it's usually not. So there's some differences between mouse and human. This is one of them. But in the mouse, normally the spleen does not support granulopoiesis. So capitalizing on that, what Norm Wolf and Trenton did was they irradiated mice and transplanted them with normal marrow cells. And then a couple of days later, within 2 days of the transplant, they did trocar implants, so basically plugs of the marrow, and they put them into the spleen. You can see that here, of mice that were irradiated. So essentially, all of the hematopoietic cells came from this bone marrow graft. And what they found, and it was really quite nice, was that in the areas where there were only spleen, the cells were predominantly erythropoietic. In the areas of the bone marrow, the cells were predominantly granulopoiesis. And then in the areas of the interface between these two organs, there was a mix of the cell types. So that was also another example of microenvironments being specialized in different organs, and also in the marrow, we know there are actually different regions supporting different types of hematopoiesis. So, and it's a bit more complex. So this is a very nice review, actually, in Cell Stem Cell. I'm the senior author on it, so I'm going to do a bit of promotion here. But what we've done is we basically summarized the use of different Cre-transgenic mice used to identify hematopoietic stem cells as well as microenvironment cells. We discussed the positives and the negatives of the different models, and we've also talked about imaging technologies that have been really useful in understanding how the microenvironment's supporting hematopoiesis. So if you want to read it, it might be a bit biased, but it is a very nice review. I'm very proud of it. So we know that numerous non-hematopoietic cells present in the marrow help us to regulate blood cell production. Now, I should say that blood cells themselves also contribute. So it's not just a one-off thing. It's not just from the microenvironment. There's a lot of crosstalk between the different cell types, but I'm going to focus on the non-hematopoietic cells today. And so these cells include osteoblast lineage cells, and I'll get to them in a little bit more, but they include arising from the mesenchymal stem cells, which are present in the marrow. So marrow contains hematopoietic stem cells and mesenchymal stem cells. And then the MSCs can become osteoprogenitors, osteoblasts, and osteocytes, which are the most mature bone cell that's embedded in the bone matrix. There's also the endothelial cells, form the vasculature. There's innervation from the sympathetic nervous system, and there are adipocytes as well here. So I'm going to simplify it for the purpose of my talk. So we have the vasculature here, and these are the CXCR12 expressing adventitious or reticulocytes, but I'm not going to talk much about them. The adipocytes, and also then the different bone-forming cells and the sympathetic nervous system. And I'm predominantly going to focus on three types: the endothelial cells, the adipocytes, and the cells of the bone lineage. So I'd like to give you a little bit of introduction into these before I go into some data from our lab. These are the three major types that people are really focusing on: osteoblast lineage cells, endothelial cells, and adipocytes. So the reticular cells, which likely form under the osteoblast lineage cells, these perivascular cells are also of high interest, as are nested expressing mesenchymal stem cells, which I'm not going to talk about as much today. In the marrow, we have two different types of bone. We have the cortical bone, which forms the outer surface of the leg, and we have the trabecular bone, which is in the inner marrow area, and it's predominantly in the metaphysis region. So it's not much further down. There's basically no trabecular bone at all, and then when you get down further, there becomes more trabecular bone as well. And these are formed by different types of osteoblast lineage cells. As I've said before, we start with the skeletal stem cell, which I usually prefer to call mesenchymal stem cells, and then they become pre-osteoblast, osteoblast progenitors. They become osteoblasts, which are actively secreting osteoid, which then mineralize the bone, and they become embedded in the bone and become osteocytes. There's also evidence that there are these quiescent bone lining cells. Not much is really known about them, and in the mice, you can't really see them until they're very old, so we don't really study them that much. What we do know is that osteoblast lineage cells, and particularly mature osteoblasts and immature osteoblasts, have important roles in regulating osteoclasts, which are hematopoietic cell-derived, and they chew up the bone. So the C, if you have trouble remembering, my little bone biology 101 is that osteoclast, the C is for chew, osteoblast, the B is for build bone. So chew up bone and build bone. So hopefully that will help anyone who gets confused. That's how you can remember it. So these osteoclasts in turn are also important in regulating bone production and the bone-forming cells. There are direct evidence using different osteoblast-specific knockouts that B lymphocytes are also regulated by osteoblasts, which can produce CXCL12, and also hematopoietic stem cells. And these studies in 2003, the seminal studies showed that if you have conditional knockout mouse affecting the bone lineage, that you actually get severe deficiencies in hematopoietic stem cells. And what does appear to be becoming the case is that the trabecular osteoblasts appear to be more important than cortical. And endothelial cells are another part of the bone marrow microenvironment. So this is the paraffin section of the bone marrow trabecular areas, and you can see all these brown stains. These are VEGF receptor 3, and this is staining up the sinusoids in the bone marrow. So it's heavily innervated with vessels. And so we know that endothelial cells have important roles in regulating platelets, and megakaryocytes, the platelet precursors, line up against the endothelial cells, and they shed their platelets through the endothelial cells into the bloodstream. There's also evidence that hematopoietic stem cells are regulated by endothelial cells, and more recently, the Frenet group have shown that sinusoidal and the arterial endothelial cells have different roles in regulating hematopoiesis, and they suggest that the arteriolar endothelial cells, which are lined by nestin-positive cells, have important roles in regulating quiescent stem cells, whereas the sinusoidal don't seem to have those roles. And also, importantly, the endothelial cells form a barrier between the developing cells in the marrow and the bloodstream, and in that way they prevent immature cells from coming into the circulation before they're ready to. And then we get to the adipocytes. So adipocytes are actually associated with loss of function of hematopoiesis. They seem to be negative regulators of hematopoietic cells, and including stem cells. A study from the Daily Labs showed this. And what's interesting is that in mice, we don't get many adipocytes actually in mice, but in humans, adipocytes become very prominent, and especially in the marrow, when hematopoiesis is actually shutting down in the long bones. So when we're born, all of the bones in our body are supporting hematopoiesis. And by the time we are late teens, early adults, the long bones are filled up with fat, and they no longer support hematopoiesis. And so basically, this red box here is where we're producing hematopoietic cells. So given that we're making billions of blood cells every day, I think it's really quite impressive. Okay, so I'm going to get to some data. This is something that I've been interested in for a while, and I'd returned from Boston and was setting up my lab and waiting for mice to come back from Boston, and I had a postdoc anxious to start on studies, and she couldn't because her mouse hadn't come back. So I suggested to her, and I had ethics to do this study, that we start working on this. So 7 years on, we're still working on it, although we're getting some publications out finally. It's been a pretty big undertaking. Looking back, I think it was a bit crazy, especially since I had a 4-month-old at the time and I had another child 2 years later. But it's been very rewarding, and hopefully it will be more rewarding in the future as well. So this was actually inspired by a close friend of mine who unfortunately died a couple of years ago of breast cancer. And I hope I don't get too emotional here. Usually I cry when I talk about it, but anyway. So having witnessed what she was going through, I realized, and I guess I kind of knew, but I didn't realize how important your blood cells are, especially in continuing your cancer cycle. So we know that most of the cancer treatments used these days at the moment, except the targeted therapies, basically kill off dividing cells. And that's okay for most organs that are fully developed by the time we're adults. But in the case of hematopoiesis, it's very detrimental because most of our blood cells are actively proliferating, and so they also get targeted by the different cancers. As a result of this, we get prolonged periods of low blood cell counts. And during this, the patient may need to undergo numerous transfusions until their blood cells recover, results in a risk of infection or bleeding, and it affects patients of numerous cancer types. It's not just restricted to those with hematopoietic diseases. And importantly, it can delay further cycles of cancer treatments, which means that the cancer cells themselves can become resistant to the therapy, and it's not a great prognosis. So when we started this, nothing was really known about what cancer therapies do to the cells of the microenvironment. And so we know that haematopoiesis is very dynamic, and we know that the bone marrow microenvironment has crucial roles in the production of hematopoietic cells. And we hypothesized that it was likely that the cancer therapies also affect the microenvironment. Otherwise, we'd expect that the blood cells would be able to recover a lot more quickly than what they do. And so by understanding that, can we improve the recovery of the microenvironment and in turn the blood cells? So what we did was we focused initially on the endothelial cells, bone-forming cells, and the adipocytes using the mouse model. We used two different therapies deliberately. These are actually also used to study stem cells in the mouse extensively. So 5-FU is a chemotherapy drug. It used to be used in haematopoiesis, and it was actually mobilizes stem cells. It's not used so much in haematopoiesis, but it's used in colon cancer patients these days, and bone marrow transplant procedures. So this is irradiating the animal and injecting them with marrow. And so we used these models, and so what we did was gave the animals a single dose of 5-FU, or we irradiated them with a lethal dose of irradiation with or without bone marrow cells. So one model, we injected marrow cells so that they would survive long term, and then the other model was a very short study because the animals die of anemia, and so they was just to investigate the impact of irradiation alone. And what we did was we did multiple cohorts of mice at different time points and included age-matched controls. They were all males because if you have females in your studies, females in different cages cycle differently, and the estrogen cycle can also impact on things like bone. So it becomes a bit harder to use females in studies like this, so we use males, although we have used some females to show that it's also happening in females. And so we did different time points of assessment. And what we took the bones from micro-CT and also histomorphometry. The marrow, we did counts and FACS, RNA, extracellular fluid. Peripheral blood, we did counts, FACS, and serum. Spleen and thymus, we did counts and FACS. And we've done these multiple times and pulled the data just to prove reproducibility. Okay, so what happens? This is basically the normal thing of what happens. The black line are the controls. You can see they're quite stable over time. In the irradiation alone, you see this drop-off of the leukocytes and the red cells, and they do die of anemia, and the platelets as well. With the 5-FU, it's known to have this leukocytosis, a rebound leukocytosis. This is what happened, and also a rebound thrombocytosis. This is normal. And then the transplant model, you can see that it takes a long time for the blood cells to recover, even though the leukocyte counts in the marrow recover really quickly within 14 days. So we look at the endothelial cells first. So what you can see is this massive dilation of the vessels occurring as early as day 2. In fact, we see it occurring within 3 hours of irradiation. So it's really very quick. And you can also see that this is accompanied by a loss of VEGF in the bone marrow extracellular fluid. So this is a potent angiogenesis molecule. It helps to keep the integrity of the vessels. And so we quantitated them, and in just the paraffin sections, you don't see much in terms of the numbers of the vessels. A little bit of a difference, but not much. Mobilization occurs here in 5-FU. Day 14 is the peak time for mobilization. But what is really profound and what we could see visually is that there's this massive dilation. What is interesting is that it recovers in both of these situations by day 14. And as I mentioned before, we observed this significant dilation as early as 3 hours post-irradiation. We actually think that's really important for a transplant. It actually makes the vessels leaky and the cells are able to get through the endothelial cells more quickly. But confocal studies were actually really interesting because they showed us something that we couldn't see from the paraffin. So if you see this is just a regular control. You get the SCAR1, which is a marker for stem cells, but it's also actually a marker for arteries in the endothelial cells, sorry, in the vessels. And this stops, oh, there we go. And then what we saw, you see this nice branching of your sinusoids, which will come back in a minute. And there's the bone there. And so you actually see that these vessels branch off towards the bone, and they're very structured. Okay, so what happens with any of the therapies, and this happened with all of them, is that we see this massive dilation, as we've seen before, but what it revealed was that these SCA1 arteries actually weren't affected. If anything, they seemed to be increased. And we lost this nice structured, I'll get that back again. We lost that nice structure. So they basically, we think they're losing signals from the osteoblasts that are making them branch out in that way. And that's all I can say about that at the moment. Although we do have some microarray studies suggesting that there are molecules important for vasculature that's downregulated in the osteoblast early after radiation. Okay, so in summary, what we get with the vessels is we get dilated and damaged sinusoids, but no obvious effects on arteries. If anything, they might be increased. And the full recovery can take up to 3 weeks, depending on our treatment time. Okay, and then we also noticed that all these adipocytes were coming in. What we did was we also quantitated them. And you can see that the numbers of adipocytes rise in all of the therapies. With the 5FU, they do restore. However, with the transplant, they don't. So they actually stay elevated over time. And this is long-term. And the adipocyte size, which is usually associated with a measure of maturation, also increases. And then it reduces, but it remains elevated in our radiation and transplant model. So that was a difference that we didn't see with 5FU. So what we were interested in, we know that skeletal stem cells, when they express RUNX2, they undergo differentiation to the osteoblast lineage. And then when they express PPAR gamma, they undergo differentiation to the adipocytes. So we're wondering if this might be occurring. So we looked at this whole marrow, and we could see evidence that RUNX2 is going down in all of the different treatments. And also notably, it never increased again in the transplant model, whereas PPAR gamma and adiponectin were increasing at early time points preceding the adipocyte formation. So then we actually used our facts-based approach to isolate out these populations. So what you do is you take the marrow and it's crushed up at the bones, and then you collagenase digest it, send it up with lineage antibodies and CD45. You take that population, you send it up with SCA1 and CD51. These contain osteoblasts and osteoprogenitors. We're actually working on further isolating these out. They are heterogeneous at the moment. And then if you stain up with CD51 and CD31, you get endothelial cells here, and then you get the skeletal stem cells here, and they can form fat, chondrocytes, and osteoblasts in culture. And so we were actually expecting that the transcripts were coming from these skeletal stem cells. And you can see that they increase over time, and that also these osteoblast osteoprogenitors increase slightly as well after therapy. But what was really surprising was that there wasn't really much happening in terms of the skeletal stem cell commitment. Slight commitment maybe, but not much. But most of these changes were coming from these osteoprogenitor, osteoblastic populations. And Sean Morrison since published that these leptin receptor expressing cells, which are contained in this population, can form adipocytes after therapy, after radiation. So that was nice. And then we also used reporter mice to determine which cell they were coming from. So we used nestin expressing cells, and we found that the adipocytes were not green, so they weren't coming from the nestin cells. We used osterix, which are more downstream osteoblast progenitor, more committed, and they weren't green. And then we used PRX1, which is a mesenchymal stem cell marker, and the adipocytes from the PRX1 were all green. So it does confirm that these adipocytes are occurring downstream of PRX1 and upstream of osterix. Okay, So then what's happening to the bone? And this part of the story is actually published. It's out, got published in JBMR last month. So I'm only going to basically summarize some of the results. But as I mentioned before, we have two types of bone. We have the trabecular bone, which we measure here, and the cortical bone, which is virtually absent of trabecular bone, and you can measure them using micro-CT. And what you can see, and these are representative pictures in the areas of the trabecular bone, is that after the irradiation transplant or 5FU, we see significant loss of trabecular bone, but this appears to be specific to the trabecular as the cortical bone actually regenerates over time. And what we have shown is that this bone loss is due to increased osteoclast activity. We were looking for the reason why the osteoclasts were increased, and one of the hypotheses was that rankligand, which is an important osteoclastogenic factor, is stimulated by inflammatory cytokines. And we thought that the damage occurring after the therapies would stimulate inflammation, which it did. And what was really interesting was monocyte chemoattractant protein 1, MCP1, was incredibly highly elevated in the animals' early time points after the therapy. And we think that this is more important than any of the other inflammatory cytokines. What MCP1 does is, as its name suggests, it attracts monocytes to the area of injury. In this case, it's the bone. Monocytes are the precursors of osteoclasts, and we think that then they form osteoclasts. Okay, so what we did was we gave mice the bisphosphonate zoledronic acid. This is an anti-osteoporosis drug. It's used in the clinic. And what we did was we gave this to mice 3 days before the therapy. And what we could show that mice that were treated with zoledronic acid 3 days prior to the transplant had completely normal bone, whereas those with the cell line, we observed the bone loss as before. And this is actually an important finding because in the clinic, bone loss in patients who are transplanted, especially allogeneic transplant patients, is a major long-term side effect, and they haven't been able to work out the proper timing of when to give these patients zoledronic acid. So we have a clinical trial at the moment because we believe that actually giving it prior to any therapy at all is going to be the most effective. And importantly, zoledronic acid was not detrimental to stem cells or to blood cell recovery. So the effects of the cancer therapies on bone, we get significant and persistent loss of trabecular bone in all therapies. We get altered bone remodeling, increased osteoblast surface, increased osteoclast surface and numbers, but this outweighs this. And it likely occurs due to increased inflammatory cytokines and rankligand. And in the clinic, the patient becomes more susceptible to bone fractures, and it occurs, and so, as I said before. And what was really interesting is that we found that MCP1 was highly elevated in serum of patients undergoing an allo transplant. This is a transplant where they get donor cells from another person. And what was even more interesting is that when we separated the cohort due to those patients who had their last round of chemo more than 100 days prior to their transplant, and those patients who had their last round of chemo less than 100 days prior to the transplant, the ones that had chemo less than 100 days prior to their transplant experienced even more bone loss that was significant at the hip, and MCP1 completely correlated with the bone loss in those patients. So that was really nice for us. So in summary, with the niche, and I'm going to get into the stem cells, I've got a little bit more time, good. What we find is that cancer therapies profoundly alter the content of cells of the bone marrow microenvironment with the cells we've investigated so far, the endothelial cells, the bone-forming cells, and the adipocytes. Major similarities are transient damage to the sinusoids of the vasculature accompanied by vessel dilation. We get significant increases in adipocytes, which is permanent after transplantation. And we get significant irreversible loss of trabecular bone due to increased osteoclast, and this is also a known side effect in allogeneic transplant. So what is the long-term impact on stem cells and progenitors? As I mentioned before, we assess these at all times. What was really interesting was that when you look at there's a few different ways to measure stem cells immunophenotypically. So one of the populations that we use are the lineage negative, KIT positive, SCAR1 positive, CD150 positive SLAM HSCs, and the multipotent progenitor cells are CD150 negative. Now, the reason people started using this is that CD34, which is used to distinguish between long-term, short-term, and multipotent progenitor cells, is known to actually be deregulated in non-steady state conditions. So people were looking for markers that could more accurately predict stem cells in these sorts of populations. So Sean Morrison came up with CD150, which we call SLAM, but what is actually quite ironic is that people are now using CD34 as well as CD150 because CD150 contains a mixture of long-term and short-term repopulating cells. So it didn't really resolve the solution, and we actually use both populations, and we find that CD34 is actually really more reliable than people think. So what was really interesting is that when you use the CD34 and CD135, which is the receptor for FLAT3 ligand, to isolate out long-term, short-term, and multipotent progenitor cells, what we see is that the long-terms essentially are unaffected. And that actually now fits in with Frenette's discovery that the arterioles, the SCAR1 positives, which we find are not effective, are important for long-term repopulating stem cells. But what is interesting is that we do get reductions in the short-term repopulating stem cells, which explains these differences here. But they're essentially normal. This is not significant long-term. But the most striking effect that we get are these significant reductions of multipotent progenitor cells, and they don't recover at all. So this was actually quite controversial, I guess, at the time, because everyone thinks that long-term repopulating stem cells are the only thing that repopulate animals. So what we did was we decided to assess the repopulating potential of these cells. What we took was our mice that were either the cell line treated controls, age matched, and mice that were 5FU treated with irradiation and transplanted. And then at 16 weeks post-therapy, we quantitated HSCs and MPPs, and we transplanted cohorts of lethally irradiated recipients with equal numbers of donor or competing bone marrow. And then we assessed donor reconstitution at 16 weeks. So what we found was that there was a significant impairment in repopulating potential of these cells, even though their stem cell numbers, the long-term stem cell numbers, were normal. And this was not due to aging. So aging basically results in a reduction in B cells and an increase in granulocytes. And so we see some evidence in 5FU, but it's not significant with their B cell reduction. And the granulocytes in the irradiation or transplant don't increase at all. So it's not consistent at all with that. So then what we did was we took the donor repopulating cells here. We plotted the donor repopulating cells versus the number of the long-term stem cells, short-term stem cells, or MPPs, and determined if they correlated. So for correlation studies, you're looking for an R-value of close to 1 and a p-value of less than 0.05. So the long-terms didn't correlate with the repopulating potential. Neither did the short-terms, not quite, but the MPPs did, and this was based on CD34 and CD135. So then we looked at the SLAM markers, and so what we found was, again, the SLAM HSCs didn't correlate, the SLAM MPPs did, and then the SLAM HPC1, which were a more mature population, didn't correlate either. So the only thing that correlated with the impaired repopulating ability was the MPP numbers. So then to further determine if this was the case, we took 5FU marrow treated animals versus saline treated. Now, there are two reasons why we did this instead of using the transplant system. One is that for all of our studies, we've used black 6 mice, C57s, as our host. And that's because the Li51, which you guys call, it's actually CD45.1 or PTPRCAs or whatever you want to call them, but Li51 is a bit old terminology, people. I mean, don't use it, please. Anyway, so the problem with the Li51 mice is that they actually do have reduced numbers of stem cells. It has been shown, and we agree with that as well. They have a slight difference in their microenvironment. So what we did was we took mice and cell line treated them or 5FU treated them, waited again, quantitative HSCs and MPPs. We transplanted equal numbers of donor and competing marrow as before, or we fax isolated these SLAM HSCs and competed them against 1/10⁵ marrow cells and assessed donor reconstitution at 6 months post-transplant. So this is a very, very rigorous test of repopulating ability. So what we found again was that the repopulating ability was impaired in the 5FU-treated marrow. But when we took HSCs from the mice on the same day and transplanted them into recipients, what we found was that they actually repopulated just fine. There was no difference at all. And then when we looked at the numbers of the MPPs using both methods, again, they were significantly reduced and the HSCs weren't. And then what was really nice is that I presented this talk at Keystone earlier this year, and when I put the abstract in, I thought, oh gosh, people are really going to be this is a bit controversial, but then these studies came out just in time suggesting that in steady state conditions, the MPPs are actually the drivers of haematopoiesis. It's not the long-term stem cell. So then what we've done to try and get at the transplant model is we've started using confetti mice. And I think that, I know that Jane Visveda's group are using these mice. They're actually pretty cool mice. They're reporter mice. They have 4 different colors, although if you have homozygous knock-ins, you can actually get more than 4 colors. What happens is the Cree recombination can give you either GFP, YFP, RFP, or cerulean. And so the GFP is nuclear, the YFP is cytoplasmic, RFP is cytoplasmic, and the ceruleum is cell surface. And you can see here from the intestine what these colors look like. There are some limitations with these mice. If you have a constitutively active Cree, they can flip. So the colors can change all the time. So it's only if you treat them and then remove them that they're useful for studies like this, which is what we've done. So what we've done is we crossed them to the HSCL Cree inducible mouse model, transgenic. These mice were generated in Glenn Begley's lab. I think that some of you are using them, not all of you though. They're actually, SCL is expressed in long-term, short-term, and also expressed in megakaryocytes and some CD8 T cells we find. So what we do is we tamoxifen treat the animals with food, and then at 4 weeks after, when they're 12 weeks of age, we euthanize them, do fax analysis, and transplant marrow from them, this whole marrow, 5 million cells per animal, into recipients, and then we let them go for 6 months. And then we also look for the confettis. And this is what it looks like by fax. So this is GFP here and YFP here. So on the Fortessa, you can have nice filters. It takes a bit of getting used to with the compensation, but you can compensate them quite nicely. And then RFP here, which is dominant in this animal. You can see here it's very strong in cerulean. So what we did was we had 2 separate donors, and then they had cohorts of animals, 4 mice each. And what you can actually see is that this is the pre-transplant. This is what the output was like. So these are the different colors. They're also a very similar trend in that they're increasing from the long terms to the short terms to the MPPs, and in both of these animals. This one's a little bit different, but in general, the trend is that they increase. But what's really striking is that after the transplant, the trend goes to an increase in short terms, but then a drop off from the MPPs, which is consistent with our finding that there's something wrong at the MPP stage. It's not the short terms or long terms, but it's happening at the MPP stage. So then if you look at the total number of marked cells, so these are all of the confetti-colored cells, and you divide the short-term HSCs, if you look at the output, dividing the short-term HSCs by the long-term HSCs, what you can see in the different colors, this is different colors, the next slide has the combined, what you can see is that the production's actually increased after a transplant from the pre-transplant. If you look at the output of the long-term HSCs to the MPPs, so if you say that the long-terms are the only cells that can make MPPs, let's divide the MPPs by the long-term HSCs, what you see is there's not a huge difference, but a slight reduction. But what's really quite striking, if you go from the short-term HSCs and you divide the MPPs by the short-term HSCs, you actually do see this drop-off in production. And then if you look at the total marked cells, if you divide the total marked cells of the short-term HSCs pre divided by the long-term HSCs pre, and the short-term HSCs post by the long-term HSCs post, there's actually not quite significant, P is 0.2. With the MPPs pre, you get this significant reduction to post, but more strikingly from the short-term HSCs. So it seems that what's happening is the long-term HSCs are fine, we're trying to make more short-term HSCs, but then there's something happening that prevents them from making those MPPs as effectively. So to summarize that, what we see is initially after therapy, we see reduced numbers of these, but then they stabilize long-term and their function doesn't seem to be impaired, at least for the long terms. With the multipotent stem cells, they're persistently reduced numbers. And we actually believe it's not an intrinsic thing, it's actually due to altered niche support of these multipotent progenitor cells. Something's happened in the marrow microenvironment and it's no longer able to support these MPPs properly. As a result, this is affecting the production of blood cells in patients after cancer therapy and in patients with different diseases, affecting their blood cells after therapy. So by understanding what's nice for us is that everyone's been focused on the long-term HSC niche and no one's been interested in the MPPs. And we think that these are actually going to be the more interesting niche to study. And hopefully in the future we can identify it and find out what's gone wrong and be able to improve blood cell production in patients in the future. So I'd like to acknowledge the people who in my lab, past and present. These are all past actually. Maria was my Swedish postdoc who actually started off. She was only in my lab for 2 years and she did a lot in 2 years. Julie then replaced her and she left last year. And Tanya, who was my research assistant, who also left last year. And then Hannah King was a Europe student who worked on this. Kobe White was a research assistant and she's now graduating from medicine. Jessica Fletcher was also a postdoc who recently moved to the Brain Center. Anna is a current RA. Chaco recently completed his PhD, and Lenny is a current PhD. And then my clinical collaborators, Dave is actually at the Royal Melbourne Hospital now. Paul Neeson and Simon Harrison have been fantastic with the clinical studies, as has Peter Ebling, who's now at Monash. We actually did the confocals with Les Silverstein's group when Julie was over there learning how to use the laser scanning cytometer, which is a really beautiful machine that does facts on a slide that we're using to further our studies now. Christos also is a good friend of mine and collaborator, and she's actually working with me on identifying the MPP niche. And then for the funding, NHMRC, Leukemia Foundation, the Swedish Research Council, the ACRF, and the Ian Potter Foundation. Thank you.
Thanks, Louise. And questions?
Yes, so I might have missed this, but functionally, is there a reason why the long bones fill with adipocytes?
I have a hearing impairment, sorry, and Matt's going to repeat the question.
In adults, why the long bones are filled with adipocytes?
No, so there's not much really understood on that one. We know that in humans who have bone marrow failure syndromes, their marrow becomes even more fatty than normal. So it's also a case of, and I think it's occurring in the mice because of the significant reductions in the marrow cells, in the hematopoietic cells, which die after the therapies, and they are space fillers. I do actually think they're more of a space filler than having an active role in suppressing hematopoiesis. And then something might come along and then reduce the adipocytes, like what we see after the therapies again, but not much is known about that at all.
Just related to that, in your studies post-therapy, you showed that adipocytes were really increased.
They increase, yeah.
Do you know if that's suppressing hematopoiesis in that?
Well, this is a funny thing. So we actually use the adipocyte inhibitor that George Daly used in his lab. And we found it had no impact at all on the repopulating ability of the marrow after the VADS treatment. It does reduce the adipocytes that form, but it doesn't get everything. So whether or not there's a particular adipocyte that's not requiring PPAR gamma to form, or whether or not, because BADS itself, one thing that they didn't say is it doesn't dissolve very well in the saline, and they use saline, so we use exactly the same thing, and it was horrible to dissolve. We finally got it to dissolve, but we don't know if that also affected the outcomes. We do see reductions, but I'm not so sure, and there are other studies coming out now suggesting that maybe adipocytes are not as potent, although the fact that we still see them increase in the transplant model, I do think that they are taking up some of those perivascular cells, and I think that they may be important for the MPPs, but that's something that we're looking into now. So it's hard to say at the moment. I don't think they're as potent as people believe, but it might be that the lineage commitment has removed the niche cell that otherwise would be supporting the MPPs, and maybe now it's a fat cell. And so that's something that we're looking into.
You mentioned that the estrogen cycle can affect bone. Does this have any clinical significance that you know of in terms of men versus women undergoing chemotherapy?
So Matt, can you?
The estrous cycle can affect the bone niche. Does that have effects on chemotherapy in men versus women? Do you know?
Well, we actually did some studies in females for the bone study, and we found that the bone loss was even worse in the females. So we didn't do the extensive time course. It's a bit too expensive to do all of that, but we definitely did find that the females were had really significant bone losses.
Can you reconstitute the short-term to MPP differentiation by co-transferring in niche cells or depleting the osteoclast or both? Have you tried to identify which component can make that okay again?
So if you transfer in osteoclast or deplete the niche, can you in some way rescue the HSC to MPP transition?
No, so we actually, with the zalegenate treated mouse, where we prevented the osteoclasts from increasing, we actually still see impaired repopulating. So it's not the osteoblast itself that are important. We actually do think it is something to do with the vasculature, most likely the sinusoid areas, and that's what we're hoping will be the area that we're looking for, but we're working on that at the moment. Yeah.
Yeah, John?
With the zoledronic acid showed that there was a protection of the bone after the radiation or chemotherapy, but to refer back to the first part of the talk, was there a quicker rebound in the hematopoiesis?
No.
To protect the bone?
So they don't recover faster either. We looked at that as well. They had actually no impact at all on their recovery either. We did that extensively. So it's very good at protecting against the bone loss, but it doesn't really seem to have any impact on HSCs or blood cell recovery at all. And that's published in our JBMR paper.
You talked a little bit about skeletal stem cells, and it occurred to me there wasn't as much known about the factors that control the different stages of commitment and eventual production of differentiated cells.
Right.
Because presumably you'd be looking for a kind of magic bullet to support the microenvironment, similar to GM-CSF or GCSF, that are frequently used in conjunction with chemotherapy and so on.
Right.
So, are there any candidates for sort of colony stimulating factor like chemicals that are known about that could be used to support a patient?
Well, MPPs are defined in the animals by expressing the receptor for FLT3 ligand. And there have been some studies suggesting that FLT3 ligand might enhance recovery in patients, and especially in mice. But what then happens if you give them too much, they get a lot of dendritic cells. So it also becomes a bit of an imbalance there. What we are hoping is that we'll be able to find something better. But it might be that FLT3 ligand, we haven't done those studies yet. It's just a bit too unaffordable for my group at the moment. Whether or not that's the only thing, though, I don't know. I don't think so, because if you've impaired the niche, it's irreversible, it's gone. So you would have to constantly inject the patient to get those MPPs to continuously be produced. So I think what we need to do is understand what the niche is, how it's affected, is there a way we can restore the niche itself? And in that respect, hopefully in the future, we'll be able to restore the niche in patients as well. So that's the big goal. It'll keep me busy, hopefully, for a while.
There's much known about the cells that produce FLT3 ligand.
Not really. I mean, there are osteoblastic cells that produce FLT3 ligand, but the term osteoblastic cells is used quite loosely in haematopoiesis and even in bone at times. I mean, there are different types of osteoblastic cells, and even if you're looking in sections of the animals, they're defined in histomorphometry as these cuboidal cells that sit on the surface of the bone. But I don't think that they're all the same. I think that they're going to be very different, and that's why we're working on plaque markers as well. So in the future, we'll be able to isolate them better and also study them better, both in vivo and in vitro.
Yeah.
So, with the different types.
It has been plagued a bit by artifacts in the past, the bone marrow niche study, but I think the current tools are obviously proving a lot better for the findings. So if there's no more questions, I'll just thank Louise for some very interesting results.
Thank you.